If this is your first time on this channel, kindly subscribe and press the notification bell beside the subscribe button to get notified anytime a new video is uploaded. Thank you.
Welcome to my biology. I want to use this medium to appreciate everyone. Thank you for always being there. Thank you if this is your first time. Thank you very much for stopping by on this channel. This is my biology, where we study biology together. If you are here to subscribe, kindly do so and press the notification bell beside the subscribe button to always get notified anytime a new video is posted. Thank you very much. I want to appreciate all my subscribers. Thank you very much. Thank you for being there, and thank you that we are doing it together. And it is getting interesting every time. Thank you. God bless you. Into this class, we continue with biology practical. But what we're going to be doing today is to look at reagents that are commonly used during practicals in biology. Reagents. What do I mean by reagent? By reagents, I mean chemicals that are used. These chemicals are to verify the presence of a particular substance. The substance could be protein, it could be carbohydrates, it could be fat and oil. These chemicals are called reagents. Summarily, these reagents, these chemicals, are mostly used when we want to ascertain the presence of a particular substance in food. So we call it food test. And that is what we're going to be doing today. Thank you very much for being there. These are the reagents we're going to be considering for this period of time. To test for the presence of starch in a food substance, iodine solution is the reagent. This iodine solution, when applied to a source that we suspect that probably there is starch, if starch is truly present, the food substance turns immediately to blue-black color, and that confirms the presence of starch in that food. When you want to write a report for this experiment, it is most of the time written in a tabular form. The first column is the test. The one in the middle is for observation, and the last column we have the conclusion, which is also known as inference. So it is written in this form. And normally under the test, the instruction has been given. What you are to do, the steps you are supposed to follow, has been stated clearly under the test. So most of the time it does not carry marks. But observation and inference, that is where candidates earn marks or students earn marks. Under observation, it is required of you to look intently into those things that you are doing. At every point in time, there could be color change or color changes. You are expected to observe that so that you can write it under your observation, and in the long run, your conclusion will carry out whether what we intend to see or what you are trying to verify is in the food substance. So let's start with iodine solution, the first reagent to ascertain the presence of starch in the food. The food substance could be tubers, as we have in cassava. It could be yam tuber, and you may be given grains, such as maize grain. If you are given tuber, maybe like potato, it is very easy. Just get your knife, cut it into small pieces before you apply iodine solution. The surface of the tuber is usually plain, but immediately you apply iodine solution, the surface turns to blue-black. So there is a color change, and that is a positive result. Therefore, in your conclusion, what you are expected to write is that starch is present. Now, what happens when you are not given tubers? You are given grains like maize grain, dried maize grain, and you have to test for starch in it. What is expected of you is either to cut it into two, after you might have soaked it in water for some minutes, or you crush it as the case may be. Then you place it on white towel, or you place it in a petri dish, or whatever the instruction given to you. Then you apply a few drops of iodine solution. Immediately you do this, you discover that the maize grain, the crushed maize grain, will turn blue-black. And that is an indication that there is starch in it. Alright, this is how you are expected to write your results in biology practical when you are testing for starch. Let's assume that in a specimen given, if it is the one that you're going to crush, like the maize grain I said, the instruction would have been given to you that you should crush it, and whatever they ask you to do, you do it. Then add iodine solution. The next thing that happens, that is under observation. What did you see? The piece of that substance or that specimen must turn blue-black. Not that it will turn blue-black. No, it turns blue-black. And between the blue and black, there must be an hyphen to show us that it is two colors that are combined in one. And then finally, the conclusion or the inference is that starch is present in it. Anytime we have blue-black coloration using iodine solution on food substance, that food substance contains starch. Thank you very much for watching. The second test we're going to be doing is test for simple sugar. Test for simple sugar. Don't forget that simple sugar are three. We have the glucose, we have the fructose, and we have the galactose. From any source of food, if you want to test for simple sugar, most of the times, Benedict solution is the reagent that is needed. And if you're not using Benedict solution, if you're asked to use Fehling solutions A and B, be very sure that you are testing for simple sugar in that food substance. Now, Benedict solution color is blue, and it is a whole reagent in itself. But Fehling solutions A and B, they are 2-in-1 solutions. Solution A is blue, solution B is colorless. At times you may be given glucose drink, given glucose powder. It is required of you to make it into solution. At times you may be given fruit. When you are given fruit, it is expected of you to extract the juice. All of this will be stated in the instruction that will be written under the test for you as a candidate. Now, if it is glucose powder, you have to dissolve it in water and get maybe about 1 cubic centimeter of that solution into a test tube. Then you add few drops of Benedict solution, and then you are required to boil. When you are boiling, most of the time it is safe to use water bath. What do I mean by water bath? You place water in a beaker, and then you heat the water. So indirectly, the water will boil whatever you have in your test tube. The color will start changing from colorless. It's going to change into green. It will change to yellow. And finally, it will change to orange. And you may see some of the textbooks, they will write orange red, because that is between orange, very deep orange, and light red. But there is provision in the marking scheme for all these color ranges. If you write orange, you are not wrong. If you write orange red, you are not wrong. So for testing or for verifying the presence of glucose or simple sugar from a food substance, we use Benedict solution. And if it is from fruits, don't forget that you have to extract the juice, put the required amount in a test tube, add few drops of Benedict solution, put it in water bath, then you heat or you boil, and note the color changes. By the time you want to write your conclusion, it's going to be simple sugar is present. I want to believe that you understand. There is no magic in it. Anytime you are asked to use Benedict solution for food test in biology practical, and you have orange as the end color of whatever you are boiling, simple sugar is present in that thing. Whether fruits, whether other sources of food. If you're not using Benedict solution and the examiner has given you alternative, which is Fehling solutions A and B, you first add few drops of solution A, followed by solution B, and then you boil. It's going to give you the same result. What we have in Benedict solution is the combination of Ferlin solutions A and B. I want to believe that we understand that. Really, this table summarizes tests for simple sugar. So any source of simple sugar given, just add few drops of Benedict solution, then you boil using water bath. If there is no provision for Benedict solution but you are given Ferlin solution, and it's going to be two solutions, solutions A and B, they must have been labeled. So you add them and then you boil accordingly. Then you note the color changes. But finally, the final color is going to be orange, and that gives you an evidence that there is simple sugar present in that food substance. Now the next food test we're going to be talking about is on protein. Protein. Yes, protein. The one that builds the body, that repairs worn out tissues, that enhances growth, especially in young children. That is what we are talking about. How do we verify the presence of protein in a food substance given to us? We have different types of tests. We have Burette test, we have Milon's test, we have Xanthoprotein test, but in the course of this class, we're going to be talking about just two. We'll talk about the Burette test and we'll talk about the Milon's test. For Burette test first, the Burette test is for protein. For any suspected protein source, the first thing you do is to get it into solution. When you make it into solution, add few drops of sodium hydroxide solution first. Add few drops of copper 2 sulfate solution. Let's take for example, you have milk, powdered milk. It is expected of us to first dissolve it in water, 1 cubic centimeters of this solution, and that is what we're going to be working with. So we add few drops of sodium hydroxide first. Check, do we have any visible reaction? Because that's part of what is expected of you to write under observation. Then add few drops of copper 2 sulfate solution, which is blue. Sodium hydroxide is colorless, but copper 2 sulfate solution is blue. By the time you add, you are not boiling this. This is not to be boiled or to be heated, but you have to shake vigorously in order to acquire uniformity in the color. And by the time you shake very well, the color it gives you is purple. This color is purple, and that's a positive reaction. That is a positive result. It tells us simply that there is protein in that food substance. If you're not using milk, you want to use maybe egg white, there is a way you extract the albumin, that's the egg white, so that you don't have it mixed with the yolk. When you get that, it is a solution, it is solution already. Just put few quantity of it into a test tube and add few drops of sodium hydroxide solution, followed by copper 2 sulfate solution, which is blue. Then shake vigorously. You will discover that it turns purple. You can also get it from stock, the water with which you boil meat. Just get a little quantity of that, put it in a test tube, add few drops of sodium hydroxide, followed by copper 2 sulfate solution. Then shake very well. Shake very well. You will discover that it turns purple. And that is Burette test for you. I want to believe that you understand. much for still watching and learning. The next practical we want to talk about is the Millon's test. The Millon's test is also for protein, and the name of the reagent used is Millon's reagent. Millon's reagent. This solution is colorless. Is colorless. Colorless means that it looks like water. Like I said, Millon's test is also for protein, and the reagent is Millon's reagent. To any given or suspected source of protein, proceed to get solution of that suspected food substance. From the solution, you take little amounts, then you add few drops of this Millon's reagent, put it in a test tube, and then you boil using water bath. I would advise that in the process of boiling, try to cork the mouth of the test tube with cotton wool, or if you have a stopper. The reason is that the content of the test tube will try to pop out. By the time you start boiling, if you are using milk, you discover that the milk coagulates, and after coagulating, it starts changing color gradually from the milk color it has into pink, and finally it gives you brick red coloration, and that's a positive reaction, and that's a positive result of Millon's test. It gives a brick red coloration. Practical we're going to be talking about is for fats and oil. Under fats and oil, we're going to be talking about just two food tests, and the first one is translucent test. It's only fat and oil that is capable of causing translucency in paper. What you need to do is to apply few drops of vegetable oil or any source of oil. It can be butter, it can be margarine, maybe asked to rub meat, boiled meat, on white paper. What you're going to do thereafter is to view the paper against source of light. You can just raise it up and view against the sun. What you will discover is that the portion of oil on that white paper will look lighter than other parts where the oil did not touch. So the looking lighter of that part is the translucency that we're talking about. So it is only fat and oil that is capable of causing that on paper. Is that taken? Anytime you are asked to rub oil or to put oil on paper, whether filter paper or white paper or any paper, you will discover that it's going to translucent. And this is practical that we do almost every day for those people who love to eat akara. Many of you like to eat suya. After eating, you will discover that there are oil stains that make that part to look lucent, and that is what we are talking about. It is only fat and oil that can do that. I want to believe we understand. Any question, let it reflect in the comment section, and if this lesson has benefited you, don't forget to give it a thumbs up, share with your friends, and tell your friends to subscribe. Thank you very much. We move on to the next and the last food test we're going to be talking about in this class, and that is Sudan 3 test. Sudan 3 solution is the reagent, and it is for the test of fat and oil, to verify the presence of fat and oil in a food substance. The color of the Sudan 3 solution is blood red. To any suspected source of fat and oil in a test tube, add few drops of this Sudan 3 solution. Soda 3 solution was originally powder, so you have to mix it with water to form the solution. And don't forget that water and oil will not mix. So by the time you add Soda 3 solution to that suspected source of fat and oil, it comes up because it is lighter than water. By the time you now boil, because you have to boil, in the process of boiling, you would discover that the oil that was floating now turns black. How do you know it is black? If you check the many scores of the test tube, you will see that it is darker, very black, compared to the next layer, which is the Soda 3 solution, and the last layer, which is the water with which the Soda 3 solution was prepared. You're going to have 3 layers of solution. The first one at the bottom, very clear, followed by reddish, and then at the top you're going to have the black, which is the oil. So anytime you are given Soda 3 solution in an examination, be sure that you are to test for fat and oil in the food. And what is the positive result? The positive result when you boil it is that the fat and oil turns black, and in your conclusion, fat and oil is present in the food. Alright, in summary for today's class, food tests. If you are a student or you are a candidate, you are given Benedict solution to carry out a test. The test you want to do is simple sugar. If you are not given Benedict solution, the alternative solutions are Fehling solutions A and B. If it tests for starch, the reagent is iodine solution, and the positive reaction is blue-black coloration. If it is Burette test, that's for protein, and the result is purple. You are not boiling, no boiling. If it is Millon's test, reagent is Millon's reagent, and the positive color is brick red. For translucent test, fat and oil, Soda 3 solution, fat and oil. Thank you very much for being a part of today's lesson. We are not stopping here. Biology Practical still continues. If you are yet to subscribe, please do so, because what you're getting from here, they are things that will really benefit you and benefit others. This is where we're going to stop. Until next time that we're going to be meeting, don't forget that you have to be loving biology, you have to be reading biology, you have to be studying biology, and you keep on sharing biology.